Africa Science Dialogue, closing the gap between science and society.
Thank you very much for joining us on this podcast. My name is Ezekiel Nganga, Africa Science Dialogue. Today we are going to discuss about science religion, and I'm joined by Professor Douglas Miano from the University of Nairobi. He's also the lead scientist working on a cassava project here in Kenya and regionally. Yeah, they're working to modify the cassava to resist two viral diseases, cassava brown streak and cassava mosaic disease. So he's going to tell us more about that. Welcome, Prof.
Thank you, Ezekiel.
So to start us off, science and religion, are they inherently in conflict, or can they coexist?
Yeah, thank you, Ezekiel. And from my position is that religion and science, they actually coexist together. You don't separate man from what he or she believes in. Whether it is traditional cultures, whether it is you don't believe in anything, because I don't still believe that is true, you must have some faith in something. That is the nature of man. And science is first of all, I would look at it as a career. Then I would look at it as a point of understanding things. The way we are saying that there are teachers who are going to teach the children how to understand things, whether they are in a modern education or whether they are traditional. So you cannot separate man from beliefs, whichever form of belief. So even if you are a Christian, if you are a Muslim, if you are a Hindu, if you are like you say you are an atheist, whatever you do, that means you are saying that you believe or in something, even if you say you don't believe in God. There is the religion by itself. You can extend it to say that you are a product of what you believe in. So you cannot separate science and a man.
But we've had a lot of skepticism when people talk about science and advancement in science, and the notion about scientists trying to play God. What is then all this about?
I think to me is man marvels what he does not understand.
Okay.
And to me, when I'm a scientist in biological sciences, I may not understand a lot of physics. I may not understand many other sciences. I may not understand astrology. So to me now, I look at the one who understands those things as one who is doing things beyond what I understand.
True.
And therefore, to me, that person looks like a semi-god in the areas where they believe in. So it doesn't mean that because I don't know something, somebody has become a god. Not really. It's just your area of specialization. For example, you are in media.
Yes.
I don't know how these things are being transmitted to wherever they are going. And therefore, to me, you are a real and a good expert in this thing. And it's a science by itself. So I don't understand which part of science then can be said to be God, because everything actually is science by itself.
Wonderful, wonderful. And then you yourself, other than just being very deep rooted in religion and being a Christian and so on, you are a lead scientist in one of the first of its kind, projects to be undertaken, not just in Kenya, but globally. You are the lead scientist for the virus-resistant cassava for Africa, a genetically modified cassava. Then, how did this in any way first, before we get into the science of what you've been doing, did your your roots in Christianity somehow influence you, in a way as you undertook the project?
Thank you. As you have rightly said, I lead the Kenyan team in the virus resistant cassava for Africa project. And what we are doing together with others from outside Kenya, we've come together to try and see how we can solve one of the problems that affects cassava, and that's a disease, and seeing how we can come up with, use the modern science in solving that problem. How I came to be in here is that my interest from the time I was young was in agriculture. And I wanted to help in understand, first of all, how the plants grow. I was fascinated by when you can see a seed, you plant a seed, you wait for some time, it shoots, it comes to a tree, then it starts flowering and it starts producing. I was fascinated by that since I was young. So I wanted to go to agriculture. I didn't know. I thought agriculture is just about planting and looking at the plants, and you do what you do so that they can produce.
Yeah.
So I wanted to understand, make it to do better, to yield more. You Murphy, when you look at a plant, you look at a fruit tree when it's having fruit, they are ripening. We are interested in it, and then after that, it goes off. If it's a crop that goes for 1 year, it is normal. Yeah. So that was my interest. I didn't understand how far that can take me into science. So, and it never came to me that there could be any reason for there to be conflict, because it was just understanding things.
Yes.
That's just as like anybody else is understanding. So even my going deep into science and doing things that I do today, I never saw it as a point of having any conflict in religion, because it was just gaining knowledge. I went to school to understand. And every time I'm advancing in understanding, it does not in any way say that I'm replacing God with knowledge.
Okay.
So, and since I was brought up in the church, and I knew that there is a superior being who created the what is, and who looks and makes things to happen. So, and my understanding does not change anything. Whether I did not do that science, whether I stayed at home, whether I just did not do anything, it does not change God. It does not change nature. It does not change things. It's only me who does not change. So I don't see any conflict in my what I do and in my belief in God, because God has a place in every society. God has the supreme being has a place. Whether you want it or not, you don't change anything. So to me, my advancement does not only helps me to understand more, but does not change the supreme being who is there. Yeah.
Wonderful. And as you earlier put it, some don't understand a certain area. You tend to have some beliefs around it, but also naturally, as we have more scientific discoveries and people are realizing more, they're able to explain more phenomena that initially were very difficult to explain, and people would say this is God and so on. Then does it mean as more scientific discoveries come along, the window for God then shrinks?
That's an interesting concept that I would say. But you see, it also depends on your limitation of understanding of God. As a Christian, and I realize that the only way that I can understand my God is reading the Bible. There, I know my God more the more I read the Bible, because that's the word that He has given me. If I don't read it, then my knowledge will remain limited to what I've heard, and the little that I have read. There is also the part of where as we worship, God reveals Himself to us. And in that part, it also helps you to understand God more. So my limited understanding of God can only be, we were to go like that, saying that the more you understand things, the more you limit God. It's like you are saying is that you are taking a part of His space because you have known. It really doesn't change that. It doesn't do that. The more I understand you, the more I know you, does not change who you are. It's only my perception that changes. It's only my thinking and my seeing of things that changes. But you can also decide or depending on your focus. Because if there are things that you've not been able to do.
True.
Like now, the essence of life itself, the bringing things to life which were dead and giving them life. Apart from, for example, I know I'm a virologist,
Yes.
and I know sometimes we can come and say that something has been archived, a virus has archived for many years, and it has been inactive, and you bring it to life. So I understand how it comes to life by the fact that it cannot operate outside of the host in which it's supposed to be. It's supposed. But if my faith was limited to that small thing that I've understood, then it's my God who is changed, who is more, my understanding about my God. So there is still things that we cannot necessarily explain through science.
Wonderful.
As individuals, we are limited. And the interesting thing is that the more I run, the more I dig deeper into things,
Yes.
the more I realize how much I don't know.
You keep learning. Even at your level, you still
Yes.
keep learning a lot.
Yes, yes.
Okay.
You realize your limitations. Sometimes you feel like somebody is looking you like you know too much, but deep within you, you know the limitations.
Great. Great. So let's get back to the science work that you've been doing. For how long have you been doing this research in cassava, and how did it start?
So I started working on the cassava as a plant around 2008 to 2009. That's when I shifted my attention to specifically work cassava. The problem that I work with in cassava about looking at the viruses was a part of my PhD studies, but I worked on sweet potatoes. So the interaction of viruses in plants doesn't necessarily matter whether whichever plant it is. It is specific to the virus itself.
Yes.
And how it interacts with the host. But it is general. You can say that one who have studied on maize viruses, one who have studied on cassava, one who have studied on potatoes, and whatever it is, the understanding and the knowledge is basically the same. The only thing is that now you understand the specific interaction. Just like specific interactions with people are also different. The way I relate with you is not necessarily the way somebody else relates with you.
Very true.
So, and it's not necessarily the way I relate with you is not the way I relate with somebody else. So that does come in because of the relationship. The same thing is the relationship that help them. Either one is taking advantage of the other, or they're having what you're calling a mutualistic relationship. And in the process, they adapt how to coexist. Now, as a man, when you are looking at that plant,
Yes.
we are taking we want to have an advantage because naturally, it doesn't mean that the relationship that the virus has with cassava is actually that's part of creation and that's part of coexistence, and there are many other relationships that we are not really interested in. But we are interested in cassava because what now we have domesticated cassava for as our food. We don't want to compete with anybody in destroying it. Okay. So actually, it is a kind of a selfish way of man looking at it because, of course, we've been created by God to be the superior beings as a part of creation, and we tend to utilize the whatever God has given us. And from the scriptures itself is God gave us the responsibility in the Bible to take dominion of the word and subdue it and to use it for our own good. So, and you remember in the Garden of Eden when God created everything and then lastly created man, and he placed him in the Garden of Eden so that he can eat from the garden.
Yes.
So it's a part of us to start to eat from what God has given us. So we started from that stage and we went to hunting and gathering because people are not many. We had enough to eat and all that. Of course, some of it is a product of sin. Sin came and we had to work so that we can earn what we have. And slowly now, part of now as the population increased, we continued with the need to produce more. We moved from where we could just gather.
Yes.
We moved to where we are what we call domestication. Then we started having our own crops, and we want to stop anyone from who is competing with us. Domestication was actually creating an advantage for the plant so that it can give us the advantage that we wanted. It's
The
not,
food that is.
Yes, the food that one. So that we are saying that because I want the produce from this maize, from this cassava, from this potato, from whichever crop that you're dealing with, yes, you exclude anybody else from anything else from eating that, so that you can have enough for yourself. And that is why we are so careful that whatever we do, we don't want to destroy everything because it has a place in science. So you have to balance. The nature balances itself.
Yes.
And even when we do, we must learn to coexist. We don't want to make anything to be extinct because it has a place in nature.
Yes.
So, and combining all those is creating a balance to understand that there is a supreme being. And even as you utilize the time that you have in this world, you utilize it to the optimum possible, knowing that you are actually a steward. Whatever you've been given, including the knowledge, you are a steward, and you are accountable of what you do with it.
I understand the project is a product. The project you mentioned, the one that you did on cassava, is addressing the two viral diseases, cassava brown strip disease and cassava mosaic disease. First of all, which technology did you apply when you're doing this? Is it, and how did you do it first?
So, thank you. When cassava is not a common, is not Africa. Initially, it was not domesticated in Africa.
Wow.
Yeah, it
I
was.
actually thought it's one of the indigenous crops. So when you talk about indigenous, I think about potato and those. So it's not even.
No. The first domestication was actually in the southern part of America.
Wow.
It was brought to Africa by the Portuguese traders.
It's not even our indigenous.
Indigenous, you call it indigenous because once it came here, it was adapted. We now started selecting the crops and deciding which one fits us. And the process of selecting and making sure that the crop that we're using is adapted to us. And the tradition of how long crop is part of what we may use called as indigenous. And you see the term of indigenous is also dependent on who is using the terminology.
Okay.
Yeah, so because there are crops which were first domesticated in the region. There are others which were domesticated other parts of the world, but they were brought into the region. So cassava was actually brought. So now when it came to this place, part of it may have come with all the other organisms that were associated with it.
Yes.
But when it also, so that means that we may have some of the problems that, or not, it's not necessarily called them problems, but say some of the organisms that were associated with the crop in the place of origin, when it is being brought, it comes together with it. So that means it may establish itself together with its own, its ecosystem.
Yes.
Then when it comes and lands in a place, it finds other organisms which are already there. And once it is planted, they will come and try to take advantage of it. So some, they will not have any problems.
Yes.
Some will find that it is not good for it, so they don't have a way of being able to survive in it. Some will take advantage of it. So the two diseases that we are having, one of them, the cassava mosaic disease, it's spread in different parts of the world. You'll find it in where cassava came from in South America. You'll find it in Africa, in different parts of Africa. You'll find it in Asia, and that is cassava mosaic disease.
Yes.
But when it came now to East Africa, it found another problem. We still have not been able to establish exactly where the disease came from. This is cassava brown streak disease.
Okay.
So because it is not anywhere else in the world, it's only in East Africa. So because it's only in East Africa, for many years the disease was restricted to the coastal region. So of course, when the crop came, then it came to the eastern coast. It spread to the other parts of the Eastern Africa. The same way it went maybe to West Africa and other parts of even into the subcontinent and all that.
Okay.
So when it came now to East Africa, and it spread to different parts of the East African region. So the cassava brown streak established itself and in a relationship with cassava. And for many years, cassava brown streak was first reported in about 1930s. Right. The cassava mosaic disease was reported earlier, about 1900 or so. So when the cassava mosaic disease was everywhere, wherever you go, but
wherever you find cassava.
Okay. But the virus that causes the cassava mosaic disease is a small virus, and the recombination or the process of coming up with different strains happens very fast because of the nature of the virus. So we do have different viruses that cause the same disease. For example, in the region, we might be having about 7 to 9 viruses, depending on where you are, that causes the disease in the region. That's cassava mosaic disease. So because of that, it was widespread. The process of of looking for resistant varieties has been going on continually. And every time when we have a new strain of the virus coming in, or a new species has developed, the scientists are also coming up with new varieties that are resistant to the disease. So that has been a constant kind of battle. So the cassava mosaic brown streak disease has been restricted to the East African coast for many since 1930s.
Okay.
So, but around 2000s, we are realizing that the disease has come up to the highlands. And incidentally, for example, in Kenya, about 60 to 70% of cassava production is done in the western region.
True.
So when the disease was restricted to the coast region and it was not causing a major disease, it was not a major problem. So we didn't give it the attention that it required.
Okay.
But when the disease came to the east, the western part of Kenya,
yes,
and it went to countries like Uganda, the western part of Tanzania, and that time, now the varieties that were developed for resistance to the other disease, cassava mosaic disease, broke down because this is a new disease and they were not developed for this disease.
Mm-hmm.
So from there, initially, when the project, our interest was to see how we can be able to solve the cassava mosaic disease problem.
Okay.
But when the disease came, the CBSD, as we call in abbreviations for cassava brown streak disease, the breakdown for resistance or the effect of the virus on the cassava varieties that were available was huge. And some of the most popular varieties went down with the disease.
Okay.
So we had now to change our strategy and decide that since we have varieties that are already developed through conventional means of normal breeding, we can utilize what we have. But the cassava brown streak disease is a problem that has no solution as yet.
Okay.
So first of all, the thing that we did is to look for how can we solve this problem using any technology. And we looked for all the technologies, the varieties. Of course, when you're doing the breeding,
yes,
you sometimes get, as long as you can be able to cross the species, you can get varieties. Some could be the cassava that could be growing in the wild, that is not necessarily consumed by human beings.
Okay.
And some of them, they could be having low yields or they could be bitter or whatever because they are not selected for our use. But they could be having genetic traits that could be what you call the genes that help the plant to resist the disease. So that means that you can cross and do selection. So, and after looking at all the places that we could think for resistance, we didn't find any. So, but the good thing was that we had the modern science, what you call biotechnology,
yes,
and one of it, what you call genetic engineering, could help us to try and see, just the way we do the immunization. You get a virus and you inject into an animal, it produces antibodies, and those antibodies can be used to help in terms of creating immunity to other organisms.
Oh, the same way we immunize children.
Exactly.
Okay.
But now, the challenge of plants is that you can't go injecting every plant. Imagine you have planted millions of thousands of cassava in a field and you want to go injecting them.
It's impossible.
It's impossible. The injection, the circulation system of a plant is totally different from the one of a human being. So we are not necessarily saying that there is supply of blood that is circulating, moving around the plant. The physiology of the plant is different from the physiology of an animal.
Yes.
So one way that had come in the sciences, especially from around the 70s, is the ability to be able to transfer a part of the gene and make it to be part of the genome of and that's why genetic engineering came.
That's why it came in, okay.
So, and we decided, why don't we try this science and see if it can be able to help us solve this problem? And we tried different means, we tried different methods, and we eventually came up what we call the gene silence mechanism, where it's a normal mechanism that happens to the plant. When the plant is infected with a virus, one of the things that it does is to silence that gene, that organism that has come.
Okay.
So for those who may understand a little bit, is that if you look at the genetic makeup of an organism,
yes,
we have what we call the DNA.
Yes.
DNA contains the genetic information, the one that is translated can see. So there is genetic information, and there is what you call the genome that has the information, and that gives what you call the genotype. That is the genetic information. When it is translated to what you can see, we call it the phenotype, or what you can be able to see. So that information that from the DNA is the one that defines how an organism looks like. So if you can only add, if that plant is infected with a virus,
Yes.
the information from the organism is from DNA goes from DNA to RNA to proteins. So in that process, if there is a step, RNA, which is single-stranded, because it's only picking the information from the DNA that is double-stranded, and that information, the specific information that is needed to make the protein for phenotype, for be able to see.
For what is expressed outside.
For what is expressed outside. So in the process, if you have RNA that is double-stranded, because RNA is supposed to be single-stranded, it's only getting the information that is needed.
Yes.
You don't have it to be in duplicates, or you only need it. The information is one, so it is brought in like a single page. So if you make them to be double, nature does not want that, because does not want double-stranded RNA, because that's not how the information is supposed to be translated.
It's a deviation from the norm.
Yes. Now, when the plant realizes that, and in other organism, it starts cutting double-stranded RNA. So if you create double-stranded RNA structure, the organism cuts it into pieces, and those pieces are used as signals to the rest of the plant and to the rest of the organism, to tell them that there is an organism that there is a sequence that is not needed in them. And that's what creates those signals. Then that mechanism, those signals, we call them dicers, and it's still science that we may not go to for now. That signal is transferred now to the rest of the organism, and says that if you find this sequence, this combination, you know it is not needed, and it's cut into pieces. And what we are saying is, because RNA has been stopped from moving to the next level of producing what you can see, which is a part of the protein that is now transformed into what you can be able to see through other mechanisms.
Yes.
If you stop that process of going to the protein, that means that you have silenced it. And that's why we are calling it post-transcriptional gene silencing. So the information from the gene has been taken from the DNA.
Yes.
Through transcription.
Yes.
It comes to an RNA, and that RNA is one that is supposed to be translated to what you can see as a protein. So we are calling it post-transcriptional because transcription has already occurred. And yet going towards, now we have the RNA.
Yes.
RNA, now we create a double-stranded RNA that makes it now to be cut into pieces, and it creates a signal. It's a normal process that occurs in every organism. If it's affected if it's double-stranded RNA is created, the silencing.
The plant to have the double, when you're doing this process, then you give it an already existing one. So when the disease comes, then it becomes double. Or how does it?
Exactly.
Okay.
That's now what I was coming to.
Science.
So the process of creating that,
yes.
You remember that the virus and the plant in this case, they are still all are trying to coexist.
Yes.
So it reaches a balance. The virus is not killing the plant, but it is still surviving within the plant. So in some cases, the plant may become resistant because it has been able to keep silencing that virus that comes. The virus, by the way, most of the viruses are RNA.
Yeah.
And majority of the viruses are single-stranded RNA.
Especially in this case, when you're looking at the ones that affect cassava.
The one that we dealt now with the science.
Yes.
So since that one, now if we create a double-stranded RNA using the virus itself, we say that you have the complementary strand of a similar strand that has the complementary of the nucleotides that are in the RNA. That now, when you create that complementarity, that cuts that into pieces. So now what we did is to enhance that process by creating that double-stranded structure. And that signals the plant that if a virus comes with that similar sequences, then it attaches itself, it looks for that, it attaches itself and starts the process of silencing.
And let me just ask about how are these viruses, especially CBSD and cassava mosaic, how are they expressed for those ones who actually have interacted with cassava? How are these diseases, express themselves in the plant?
So the expression starts from within, what you cannot see.
Okay.
When a virus by nature cannot be able to enter into the plant by itself, it must be assisted to get into the plant.
Okay.
And that can be assisted by injuring the plant, and or you can be assisted by an insect. And basically what you want is, what you're saying is that that virus must enter what you call into the cell, the cytoplasm of the cell. Even if you place the virus on top of the leaf,
Yes.
it doesn't have a mechanism of getting inside. You must injure the plant. That injury can be either be mechanical or it can also be assisted. The assistance for these viruses, the two of them,
Yes.
is done by an insect known as a whitefly. So that whitefly, when it lands there, it comes and it can be able to pick the virus as it is going to the cytoplasm, into the plant cell, so trying to get the food for itself.
Feed.
It is sucking. As it is feeding, it picks up the virus, and if it moves to another host, which is healthy, it transfers the virus. It's also another process because the interaction of the virus and the insect is also very specific. And you see that you are talking about these viruses being transmitted by the whitefly and not by another one, not by an aphid, and by a beetle, and not by a mealybug. So we are saying that is specifically a whitefly because that interaction is also very.
And technically, the whitefly doesn't know it's spreading disease. It's just feeding.
No, it's just feeding.
Okay.
So, and the interaction most likely is that eventually the virus had to have a way of moving from one host to the other, so for its survival. That's a part of nature. And that's what you are saying, is that there are some things that you may not necessarily understand. Because even as you try to understand them,
Yes.
Why did God decide that that interaction has to be like that?
Only with the whiteflies.
Only with the whiteflies. Okay. So now we are coming to how it expresses itself.
Yes.
When it is picked from an infected plant to one that is not infected, that virus is deposited in the cell again. When it is deposited into the cell, a number of things happen. Number one is that this happens, it could be only a single virus particle, as we call it. That single virus particle, it has to make many copies so that it can be, it's not the only one that is supposed to spread to all the plant, because if it spreads, it will only be eating or killing where it is. So it has to make many copies. So as one of the steps that it has is that the structure of the virus is that the nucleic acid or the genome, the genetic information, is protected by a protein. So that protein must first of all be able to separate from the nucleic acid, because the nucleic acid, they need DNA or RNA, but for most of the plant viruses, they are RNA. Yes. It is now, once it is removed itself, it uses the resources from within to help itself multiply. It's a part of a biology that we may not necessarily also go beyond between here. But now, why the virus becomes a problem in management is because once it removes itself, it behaves like a part of genetic information within even the genetic information of the plant. It utilizes the same materials, the resources within the cell for its multiplication. Okay. So as it's multiplying, it has to make its own resources. So the plant starts to fight that process. They realize that something else has come. Yes. And it is utilizing my resources. And it is utilizing my resources. One step is to try to fight it. Yes. So there are different ways of fighting, which part forms now the part of the resistant mechanism of the host. Mm-hmm. But remember, all this is happening in a single plant, in a single cell. If that process of multiplication becomes successful, and there is no process, for example, of silencing that makes the virus to be cut to pieces, yes, then it matures it creates its own nucleic acid, the DNA or RNA, it makes its own proteins, then the proteins come again and cover, it's like giving a coat to the nucleic acid, then it becomes a complete virus. But that is within the cell. So from that cell, for it to be able to inhabit the plant, it must be able to move from that cell and move to the next cell and move to the next cell.
Yes.
And that process is also, the plant also restrict in some ways.
Okay.
So if it is able to restrict, then the virus may not be able to move from that single cell, and therefore the infection may not be successful.
And may not even be expressed at the end of the day.
It may not even see it.
Okay.
Sometimes a few cells might be infected, or the cells which are surrounding the cell that was infected may actually be triggered to now the cells that are not infected. Mm-hmm. So that cell that was infected dies, and sometimes you may see some plants which are infected by a virus, and that cell, the cells which are surrounding where it was infected, it may actually dry up and fall. And you see some holes, and it's what you call part of what you call a hypersensitive reaction to infection that has occurred. The virus has been restricted. But when the virus is able to successfully move from that cell, it has multiplied, made many copies. Then that moves to the next cell. And next to the next cell. That's also a process, and the plant is also restricting. There are many things that happen there, because in the process of that movement is also has a scientific, it's a form of interaction that we, for those who do the science for viruses and interactions, we explain what happens. If that is successful, then it is able to move to other parts of the plant, of what you call the vascular bundles or the xylem and the phloem, for those who can remember the biology. Then it moves to the rest of the parts of the plant.
Yes.
When that has happened, now when it has utilized it is utilizing the resources of the cell, a number of things are happening. So number one, the food materials that was supposed to be made for the plant now starts. Eventually, what you need is the energy. You remember those ATPs and all those when you are talking about the energy that is being produced?
Yes.
The other organelles which are in the cell are supposed to be using that energy. And you find that there are nucleus, there is the mitochondrion, the chloroplast, and all those. When the cell is being invaded, those organelles within the cell starts being affected. For example, if the chloroplasts are affected, then they start losing the color because the plant has now been affected. Okay. Some of the organelles might be enlarging, becoming large in size. Some of them might be destroyed by the presence of the virus. Some viruses might not be multiplying in the cytoplasm. Some could be multiplying in the nucleus. Eventually, they may destroy the nucleus. All those organelles within the cell are affected.
And for instance, when we have the chlorophyll being affected, then we see the leaves. The leaves are the ones that we're going to see turning yellow.
Turning yellow. Okay. So the turning yellow now of the plant leaf or the area that you can see, yes, is because of successful expression of the viruses and the fight. That means the fight within the cell. The viruses are actually the ones which are overcoming the processes of the cell.
And it's not old age. That's what we used to imagine when we were.
That yellowing, you see it even in the very young shoots which are coming out. So it's not necessarily because of age.
Okay.
So it may facilitate the aging because eventually, as it is yellowing and there is no development that is occurring, yes, so eventually it also ages faster and falls off because it cannot be able to be used again. But what I wanted to say is that now that successful expression of the viruses within the cell can be manifested in many ways. Manifestation is what we see. So remember, we are saying that organelles could be either enlarging as a reaction. They could also be shrinking, but they could also not be multiplying at the same rate. Those are that they could be what you call the hypoplasia. That means that the organelles are not developing at the speed at which they are supposed to be growing. There could be hyper reactions. That means that there could be enlargement of the organelles or even the cell itself. So you start seeing that there is a plant when there is a lot of things that are making plant not to grow fast. That's when we see that the plant has become a dwarf. So sometimes the cells may die because of these reactions. We call it necrosis. The necrosis means that the organelle has died. The organ has died. Then it could be what you are calling the chlorosis. That is the loss of chlorophyll, which now is expressed of the yellowing of the plant. So that successful development. So sometimes those symptoms could be seen where the virus came in. That we call them the Rocco symptoms, or we can see it now being spread all over the plant because the virus has been able to move to all parts of the plant, and that we call it now the systemic infection. So the expression of those symptoms could also depend on how the plant interacts with the virus. So you can see some of them, they are developing specific patterns and which are characteristic of those viruses. And by the way, we name viruses depending on what we see is the effect of the manifestation of the interaction of the virus with the plant. When you talk about mosaic,
Yes.
for me as a
Cassava
scientist,
mosaic disease, yes.
because the cassava, the virus is causing mosaic symptoms.
Mosaic kind of patterns.
Yes.
Okay.
You see, when those you have African print,
For those ones who did art.
similar to what even this, the background here is some form of mosaic.
Yes.
You can tell clearly where the green color is and where you can see the border between the green and the white. You
Yes.
can see the border between the blue and the white. It's kind of that's what you're calling a mosaic. There are others which may not be very specific. For example, when you talk about the mottle, the mottling, there are symptoms which you may not necessarily be able to. You can see the plant is sick. You can see the symptoms. There is yellowing, but and by some mixture of yellow and green, but you cannot necessarily tell clearly where the difference is. There are those which could be expressed within the veins. So there could be vein clearing, there could be vein budding, there is dwarfing. So the naming of the viruses is dependent on the plant in which the virus was first of all reported from,
yes,
and the type of symptoms, the way they are expressing themselves. That's why
but you can be able to see.
Yes, you hear cassava mosaic virus, cassava brown streak virus, maize streak virus. You talk about tobacco mosaic virus. All of them, you are now seeing the way they can be able to express themselves. It's by the time you are seeing what you're calling the symptoms, that means that there was a big struggle between the plant and the virus. And there's some
at the cell level.
At the cell level.
Okay.
And at the organ level and the whole system level. But it first of all starts at the cell level.
And that means the plant defense failed at that level.
Yes.
And then there is that multiplication. So, for instance, when you are looking at the brown streak, cassava brown streak disease, what are the expressions that somebody will be looking at to see, okay, this plant actually is sick with CBSD?
So the symptoms that are developed by when you have the virus infects the cassava are in different ways. You may see some symptoms on the leaves. And especially the expression here is mainly seen on the older leaves.
Okay.
You may see, and that's where you see some streaks of or the veins starting to turn yellow. But they are very fine, so it's not very clear. For one who is not trained, you may not actually tell that the plant is sick.
Okay.
But the name streak came from the symptoms that are developed within the stem. So if you look at the stem, you may see some browning streaks. So those is where you are calling cassava brown streak. You see the browning in the stem, the streaks which are looking just like lines which within the stem which have turned brown.
Okay.
But the most conspicuous expression is in the roots. So, and then there is necrosis in the death of the organelles within the root cortex.
The rotting, actually, necrosis.
Yes,
they show like they express themselves like it's a rot.
Yes, it's like a rot.
Okay.
But you can see the cells are already dead and they have become hard. They have mummified. They have the part of the plant becomes hard. It cannot be eaten. It turns brown. Most of the cassava that you have are whitish or creamish. And we've been trying to have the ones that have high beta carotene, so that means they may change color from white towards yellow. And so that means that that is increase of beta carotene, and that may change to brown. So that browning is the best expression of a cassava that is infected with the cassava brown streak virus or cassava brown streak disease.
Great. And you've very well explained from the cell level, from the gene, you're going to the building blocks of living things, And I think maybe that's where this notion about playing God and recreating, organisms comes from. Do they have any morally, first of all, is that even right?
It's God who gives us knowledge. And
okay.
We started by saying that God told us, be fruitful and multiply, subdue the word.
Subdue.
So take advantage of whatever is there. So we've been given a part of the creation. We became we were given the breath of God. So that means that we have godliness in us. And that's why we have the ability to create.
Okay.
So our understanding of how science works is because we have been able to study how the plant behaves, how the organisms behave, and you see this information is actually very recent.
Yes.
And this recent information is, for example, Watson and Crick is within the last 1 century. So most of this information of understanding of the genetic information is very recent. Initially, we started by exploiting what you can see. The science was based on of what can be observed from naked eye.
Mm-hmm.
As science increased, we were able to go deeper and study and understand more. Okay. So it doesn't necessarily mean that science has been evolving with time. So, and if that it does not mean now we are acting God because we have understood more. There are still many things that you don't understand. That the generations that will be coming, they'll be understanding and will be wondering, why were you not seeing these things? Okay. So those times when Noah was building an ark, and that humongous thing supposing to float in the water and all that, yes. Some people were wondering what is wrong with you. So science advances.
But now we have cruise ships.
Exactly. Yeah.
So, and people actually, yeah.
Yeah. And even now we have stopped just moving from what we can see on the surface and moving up. We have the planes. Somebody decided that looking at the birds flying in the air and wondering, why can't I be able to fly?
Just utilizing that ability to recreate that was given to us.
Exactly.
Okay.
So that ability to recreate is inherent in man. Okay. And continues advancing in science. And by the way, for those who study the Bible, they understand that even this was prophesied by those people who are looking in the days to come as a form of God telling us that there will be an explosion of knowledge. Mm-hmm. Towards, as we come to, we advance, and as we move maybe towards the end of the world we know it today, there will be an explosion of knowledge. And this is where we are. All of a sudden, something that started like 50 years ago, you realize that the exponential growth in understanding of this, of what is happening, yes, is moving at a very trend. You do something, and 2-3 years down the line, you thought it was a big discovery.
It was a very basic.
It is basic, and it helps to other people to
now build on it.
Yes, of the building blocks of life, what you call the nucleus, those forests as we call them, the ACTG, yes, is it was a very, very fundamental breakthrough in terms of understanding genetics. Great. So yes, and then after that, understanding how the processes work, moving us to the next level. So it doesn't mean that understanding how God did things those years, it did not change now. The same way things were operating 2,000 years ago, our understanding today and being able to manipulate does not mean that you have changed the one who created.
Yes.
It's only our understanding that has changed.
That has changed.
Yeah.
Sure.
And even if if we change the organism, the way we know it, and we say that now we have combined maize and beans and it's producing something else, yes, we cannot call it maize.
True.
So to me, there is nothing that we can call plain God, because it's a part of the process that when God created us, He told us to subdue the world.
Subdue and give us the ability to recreate.
Yes.
Now let's look at it 14 years down the line after you started research working on cassava. Mm-hmm. What are the milestones that during this process of research, and where are we with the project right now?
So the
virus-resistant cassava project for Africa, and the kind of milestones that we've had so far.
I get you. So this science is highly regulated, and it's actually the scientists themselves who agreed that we have a new way of doing things. Yes. We don't know how it's going to interact with the rest of the organisms in the environment, and they came up with immediately those early years when they started the process of being able to transfer genes from one organism to another in a way that does not happen naturally. They decided within scientists came up with a process of doing this science. You must be able to come with a regulatory mechanism, a way of governance of how science is done. So the milestones for us, especially for example in Kenya, is that we have had now that mechanism of being able to regulate. So to us as a country, and it was a good plus. And of course, for the other parts of the world, some of them are still struggling to come up with a regulatory system. Others have been doing it for many years, like the Americans. They have been doing these things for many years now. Basically, to say that let's make sure that whatever we do it in safety. And this is not necessarily only done in genetic engineering. Even everywhere you go, there must be safety rules.
Yes.
Even the planes, even the vehicles, anything that you are going to utilize, you must have a way, including the knives that are in the kitchen. Yeah. You must have a safety process of doing things. Some of them, you may not necessarily a formal school to be trained, but you are trained. The saying that says that mtoto akililia wembe mpe is actually a training process, saying that even when they want to know that fire is hot, they have to put the finger.
Let them learn.
So once they put the finger there and they burn, it creates a process of protection. So that as they grow, that frame in a candle can only be able to burn finger.
Yes.
But if they have the bigger fire, they appreciate what it means because it can now burn the whole body. So every process that we are part of regulations, when it then we have to advance, come up with those, especially for areas which may not necessarily be very and or not be practiced by everybody.
Okay.
So that to me is when we started the work, we didn't have necessarily a regulatory mechanism to regulate the science. And when the regulations or the safety law was enacted in 2009, and you can see we already started the work. NBA, the system the way it is, came in 2011. Yeah. For us, by that time, we had already seen that the science could help us in cassava.
The NBA is the National Biosafety
Authority that is supposed to regulate the GMOs. But does not mean that you're not having the regulation. Yes. Because the world had already come up with a system. So to me, that was a big milestone. So the other one is to be able to say that you can be able to use the genes from the virus itself and use it to create a system of controlling the virus within the cassava, because the plant can diffuse and not able to express itself. Yeah. So that was also a big milestone to show that you can use the technology to control the science and use it safely. That means that the plant is not affected, still grows well, it still is able to protect, and it is still able to fulfill the need of the safety aspects. So that was also a big plus, because this was the first time that cassava was actually modified. So
not just anywhere globally, because mainly it's an African food. We appreciate it more as African, when we eat it and the uses we put to it.
Exactly, and that is also critical, because you see the people are solving their problems. The people who are coming up with better ways of solving the problems of maize are the people who grow maize. The people who are coming up with better ways of managing pests in cotton, they know that cotton is...
Yeah.
So cassava was our crop. So together with now the international community, when you tell them that our crop is being affected with a dangerous virus, then can we come together and help us and assist us and participate in the process of coming up with a better variety of cassava? So we feel proud that we went this way, and at least we can say that we contributed the world and part of the science that is helping to solve some of these problems in a common crop that is common in the region.
Okay, great. So what stage are we now when we look at the project?
That's where I was moving. So there has been stages that, first of all, the science has worked.
Yes.
Then we have been able to show that the crop can grow well in the field.
Because first you start in the lab.
You start in the laboratory. You move to and make sure that everything is, first of all, start in the mind.
Yeah, of course.
So, and by the way, this is very critical.
Ideation.
Because that ideation that start process of trying to think through how you can be able to come up with a concept and move it to a process of where it can actually be actualized it's a big process, and actually it's the most important.
True.
Because now to make things that can be seen. So, and it also involves convincing other people, because we could not do this work. As scientists, we are employed by institutions.
Yes.
So means either the management of these institutions,
Yes.
the other scientists, and tell them that this is what we are thinking. And of course, there are those who can say that now this cannot work, this is not going to be safe. And so, of course, there are those things of creating God, it will be harmful to the environment.
Yes.
You must think through and say that, no, we have considered that all these things, we are utilizing, we are getting the genes from here. So that means that it is not dangerous. It is a system that is already existing.
And
so
it's ethical as well.
All those ethics and the effect on the environment, the effect on economy,
Yes.
because you don't want to come up with things that are going to make you to be isolated by the rest of the world.
True.
So moving from the mind to the laboratory, to the greenhouse, then to what we call the confined field, and moving from the confined field to now saying that it to be released to the farmers. So we have gone through all those processes. And in the process, there are many institutions that come into play. There are many institutions that if you move from one stage to the next one, you realize that there's somebody else who's supposed to overlook what you are doing. You start with the institution. The institution starts now, you start with your colleagues. You tell them and they say, okay, it is feasible.
Yes.
You go to your institution. Then you find that the institution has created institutional biosafety committee,
Yes.
a group of scientists who are coming from all sectors, from economics, from socioeconomics, from scientists, and all those, and saying that we see no problem. So then you realize that the moment the IBC has agreed, then you go to National Biosafety Authority. You persuade them and convince them, and National Biosafety Authority, now that's where other regulators come in. As you move outside now to the laboratory, to the environment,
towards the farmers' fields.
towards the farmers, and even in confined environment, other institutions come in, because, for example, NEMA will have to look at what NBA is doing.
Yes, the
But
National
now,
Environment
Management
Authority.
Authority.
Okay.
Which now, moving from there, when you come up with a variety, the people who allow varieties to be commercialized, because they have value to the farmers. Of course, you don't want to give anything to the farmers. If you are giving them which is similar to what already is available, the question is, why do we have to invest if it's not giving any benefit?
What value is it?
Exactly. So that value is the one that is now we go to KEFIS. They're looking at the variety and seeing it has advantage for the farmers to grow it. So they do what we call national performance trials. They also look to make sure that variety can be identified. That's what they look at, the distinctiveness, uniformity, and stability. So that means that you're not planting it this time and tomorrow it will be looking at something different. They have to have a way of being able to identify that variety. Once they agree that variety, and we've already done that, big plus,
yes.
We've already finished that step. Now KEFIS is looking at the information that they have collected, the data, yes. To say that out of this number of varieties that you've come up with, we had selected 8 of them. So those 8 varieties have been assessed. And now they are saying that disease, it is resistant to the disease because that was our target. They are also good yielding. They have good yield. They are not affected by other pests, and they can be grown in different regions. So that is now where we are.
So basically, the KEFIS, the Kenya Plant Health Inspectorate Service, says you have value. You are bringing in and you're adding value to the crop. There is something new about that you're bringing in.
You are giving a farmer something that is advantageous.
Okay.
Yeah. So when they give that go ahead, because this is a regulated science, the NEMA may come in and give the go ahead to say that the plant is not going to have impact in the environment. So when you grow it, it's not going to decimate other plants and all those. So then that means that you go ahead to do the planting in the environment. But the commercial release and its utilization is based on safety, what we call the risk assessment, and that's approval that is given by the National Biosafety Authority. So we are in the final tail end of the project, and now we are trying to see, once we get those approvals, we have enough materials to give to the farmers, and hopefully, and we are praying that by the end of this year, we can have the variety being available to be used by farmers.
To be used by farmers. Maybe just briefly, you can tell us 2 or 3, top advantages that these new varieties bring to the farmers.
Thank you, because this is critical. Number one, we still don't have a source of resistance to cassava brown streak disease. There are many sciences that are going on. We have there are reports that we have gotten natural resistance from some cassava varieties from Colombia and that region, the Southern America, that we say that's where cassava came from.
Originally, that is where cassava came from. Yes.
So there could be sources of resistance.
Yes.
Scientists are still working on them. We still don't have varieties that are resistant to. There are cases of tolerance. Tolerance means that the cassava is able to coexist together with the virus and produce.
Yes.
But you don't have any source of resistance. So this still remains to be the unique source of resistance to cassava brown streak disease all over the world.
The one and only available at this time.
At this time.
Okay.
So that is a big, big advantage. So that means that you grow your crop, you don't have no worry. And the problem of cassava brown streak disease is that because of the way it is expressed, remember when you're talking about the expression in the form of symptoms,
yes,
and you say from the leaves, it's not very clear. So the plant will still grow.
Yes.
From the stems, you need to have interacted with the disease a lot to see. So the plant can actually grow.
Eye.
Yes, the plant will actually grow and reach the end of harvesting. And you are thinking you have yields.
Yes.
Then you go to uproot and you find everything is rotten. So having a variety that you don't have to worry
yes,
about in between what you are coming to find it,
yes,
and you can't be coming every time to uproot the root and cut and say that you want to see.
Yeah, now this is fine. Let's continue. Let me continue waiting.
Yes. So that is a big, big advantage. Mm-hmm. So the other advantage is that we combined now the modern science that is giving us resistance to cassava brown streak disease with natural resistance that was there for cassava mosaic disease. Mm-hmm. So we are giving a variety that has double resistance to cassava brown streak disease,
yes,
and resistance to cassava mosaic disease. So the two are now, you are getting a variety that you can plant and you don't have worry that your yields are going to be lost because of that. That is number one. Yes. Then number two, of course, we have assessed for our other biotic strains that could be the pests and diseases. Mm-hmm. And the other thing is that these varieties are also of high quality. They are good yielding. Okay. So that means that they may yield higher than the varieties that are there.
What is the tonnage? What are you looking at? What are we looking at when we talk about high?
So the tonnage that we have in Kenya now is at about 9 to 10 tons per hectare.
Okay.
So these varieties are giving us 30 tons per hectare and above. By the way, the other countries are getting about 18 to 20 tons per hectare.
Okay.
So in the African continent, because of being in the tropics and having many challenges, yes, we are producing at around 9 to 10 tons per hectare. But you can actually be able to, on average, we can actually be able to produce at double that.
So with this new variety, we're saying we can do 3 times?
3 times, yes. Wow. On average, we can do 3 times.
Wow, that means a lot to the farmer in terms of food,
yes,
economic empowerment.
Exactly. Okay. And mainly because they are resistance to that disease. Brown streak and cassava mosaic disease can actually destroy the materials up 70, 80% or even to 100%. Wow. So that means you can go and harvest nothing.
After a year. Cassava takes around...
It takes 9 to 18 months, depending, but on average, most of the varieties takes 1 year. And actually, we say that the average for cassava takes 1 year. Okay. Imagine you have invested, you have planted, you have plowed your yard, you have planted, you've invested in seeds, you have weeded, you have waited for a whole 1 year, and you come and find nothing.
Nothing. When you uproot, you can't take them to the market, you can't eat yourself.
Exactly. And then, so we have better resistance, having you have advantage in the yield, either because you have reduced the losses or the varieties are producing much more than the others. Yes. And then also the quality of the roots is some of the quality of the roots are much, much better than what you've already been there. But that was not, our target was to maintain what the varieties that the qualities that the farmers already had.
Yes.
And because our target was farmer preferred, but now we are realizing that commercial aspects are also very, very critical because as we industrialize,
Yes.
we also need these cassavas to be used for industry. So huge advantage. And there is a chain of that. If you have better yields,
Yes.
it means that many other advantages come in. It's a trigger because the critical thing is that let's have the yield.
Yes.
And once you have the yield,
once you have a surplus of the yield,
once you have enough for subsistence, you have a surplus, then you start kicking in many other benefits that come in.
Yes. Yes.
And of course, we are expanding our industry. We are expanding our science. We are expanding our capabilities. So there is a ripple effect of when you can be able to show that you have a system of utilizing a science.
And now when we talk about systems, cassava ideally or normally as we grow it here and in many African countries, the seed, it's propagated through the stems, right?
Yes.
And ideally the way they do it is I go to my neighbor and tell them, I like your crop. I like the way it appears. The stems are big. It's greener. Can I have a piece? Can I have a seed? So how are you going to have to ensure that these new varieties that you're introducing, you are actually going to be able to not mix them up? Because how are you going to make sure that you can be able to identify that this is the improved varieties? Can go and say this is the improved variety. So I want this.
So important question because it's a part of what we call coexistence, and we actually want to hope that we can move away from that system of borrowing.
Okay.
Because the system of borrowing is kind of a subsistence form of doing things.
Yes.
And if it is only for food, it makes sense. If it's for commercial purposes,
yes,
you would want to have a good quality of seed.
True.
So, and then you'd want it to be produced in a system that that's giving you the best. So, and the importance of seed in crop production is very, very critical.
Very.
So, so that you can start with good planting materials, the major institution in Kenya that is leading in this work is the Kenya Agricultural and Livestock Research Organization. That is the premier research institution in the country, what we call CARO.
Yes.
So CARO is the institution that is leading in this work. CARO, they also have a seeds unit.
Mm-hmm.
And initially, it was looking at especially the crops that what you used to call the orphan crops, the crops that many businesses may not be interested in producing seed for the farmers. Because this is a national, they created the seed, at that time they called it a seed unit, so that they can be able to produce the seeds for the farmers of high quality. And so then they have low disease pressure and all that. Then they can be able to give the farmers what is of good value.
Okay.
So when the crop is planted in the field, it's exposed to many factors. Virus diseases is one of them. So when it goes, it will be affected by fungi, it will be affected by bacteria, it will be affected by all manner of things which are living together. That may lower the advantage of the crop. So I want with a healthy seed, and that's the responsibility. When CARO is producing the seed, the other organ that comes to see the quality of the seed that was produced is KEFIS. Because KEFIS deals with seeds. They're the ones who are coming to say that, yes, this seed can be used and we have certified that it is of high quality and we can be questioned about it.
Mm-hmm.
So the seed that is produced, we are creating a system, and most of what you have described now is what you call informal seed system.
Yeah.
It drives the process, and it have to be there.
Yes.
And we may not necessarily eliminate it completely.
Okay.
But if you want to do cassava production for commercial purposes,
at large scale mainly.
At large scale mainly. Mm-hmm. So we have to have good sources of seed.
True.
And that seed must be in big amounts, not the one that you're going to get two cuttings.
Two cuttings.
So what we are creating is to create a seed system for cassava.
and be guaranteed that the seed system I plant at the end of 9 or 12 months, I will actually harvest good roots.
Exactly. So we are creating a seed system. Caro is reading that. We have other donor, other partners who are coming in to help, especially the scaling up and creating a seed system, so that then when you are in a particular region and you wanted our seed, we can tell you, go and get, if you say you wanted 10,000 cuttings.
And you are probably within the coastal region of Kenya.
There is someone where you can tell you, go and get the seeds from so and so.
Okay.
So we are creating a seed system, and this seed system for cassava is going to help us to maintain the quality, to maintain the numbers, and also to be able to know that we can be able to know who is growing and can have a track. So how do we know our varieties? We have a system, and that's why DUS, the durability, uniformity, and stability, is done so that then you can be able to define the variety. So it has unique characteristics, and you can be able to tell either using the modern sciences, so that you can tell that there is the presence of the gene in there that is protecting. So you can be able to test for that. And even when you look at the variety, we can be our variety is this one because it has these unique characteristics. Just the same way when you go to look at the maize, the people can tell you that I've planted 511 or the 5 series
or 614.
614, and they'll look at it and say that yes, this is 614 or this is Katumani. Those are the descriptions that we are saying that. So you can go and say that yes, this is our cassava.
Great. And the whole process that you've described to us, and you told us that you went to the point of looking at the quality of the roots. Have you had a chance to, quality of the roots, one of the ways to determine them is taste, How have you looked at, have you tasted? Do they taste nice? What can you tell us about that? If at all you have done that, of course.
Yes, we have, because it's a part of testing and because of as a part of research. And after we were given we have been able to know that it is safe and it's good and it's everything else. You can't take to the farmers what they have not, you have not known how it tastes.
Yes.
So at a limited level, yes, we have done everything to confirm that it has all the qualities that it has. We are ascertained safety, we have ascertained everything. We've done the record testing as scientists to confirm that, of course, after we have been given the government has given the consent that it is good. But this is at the level of where we are saying that it is as scientists who we are testing to see cook it and feed it, see how long does it take to cook. Not so much of many other things, but especially the basics, the way we do it at the kitchen level.
Things that would matter to the farmer,
to the common consumer. Farmer, the consumer now who is consuming cassava. Yes, we have done the cooking. We have seen. We can tell you this variety takes 20 minutes. When you are boiling, this takes 25. This takes 30. When you look at it, when it has cooked, it is flowery or it looks grassy. You can tell how it looks. You can taste and tell whether it is. It has some bitter taste or whether it has sweet, and almost all of them have some sweet, and sweetness, not the one for sweets.
Okay, just some level of.
We define how we define the taste of the cassava. All those qualities are actually defined by the others. So we do have how they have all those qualities are.
So would you say they are sweeter than the others? They are better in terms of the composition,
This is relative because it also depends on people.
Yes.
And so for where we are coming from and the qualities that we know that farmers look for, we compared them with local varieties. And of them actually, and then what you do is that we rank them. So we rank them brightly. So you have you have boiled these or done whatever you have done, and then they have been labeled by somebody else who is independent and comes and calls number 1, 2, and 3. Then once you test them, you come and ask. So among them, which do you give the scores? And those scores help you to come and you add them up, and you have looked at different characteristics, and then you can add those scores and see which one has the highest score.
Okay.
But even
so that means you at this stage you don't just have one variety, you have several varieties.
You have several varieties.
Okay.
You come and compare them so that, especially to answer of whether they are better than what you have.
Yes.
Then you come and, of course, the first thing that you're looking for is that to confirm that you've maintained the quality of when you started, because the farmers have already what they like.
Yes.
And if they have what they like, What if you bring other qualities? You don't know if they selected them against those things that you are thinking that they are good.
Yeah.
So the best measure is to see that all the qualities are similar to what is available.
Maybe can you describe to us the one that, because you tested these ones,
So
what stood out for you in the variety? As much as you are the lead person in the project, and of course you're biased somehow, but you have several to select from. Which one you can describe to us? How the one you felt is much better? How did it feel like? Generally, what was your feeling about testing your work after 14 years?
Exactly. So when you look at them now and you're comparing them, of course, after you have then you do ranking, and you say you rank number 1, you rank number 2, and the scores are added and all that. Surprisingly, that out of the 8,
Yes.
4 of them scored slightly higher than the most popular variety.
Wow.
And this was huge to us. Actually, then you are feeling like, oh my God, this is what I've been working for all these years,
Yes.
and this is what I've been able to produce. You feel like, oh, thank God, because you are looking for it. Is those moments. You young people, you call it the wow time,
The wow moment.
The wow moment. So yeah, it was so fulfilling. Actually, I say now that at least for that, I feel I've contributed to the community in a big way. So, and when the scores came and you have been ranked them, and you see that in science, we measure, not necessarily saying that because you are 1 centimeter taller than me,
Yes.
to mean that you are taller.
Yes.
We say that you are significant difference. So
Yes.
they're all, which was critical to us, because, but you can see that if you are given the majority of us, there were the scientists, we actually felt that we actually go for this.
For the improved
yes,
variety.
for the increased, actually 3 of them were ranking slightly better than the ones that the most popular of the varieties that.
So technically you can confidently confirm that as it is right now, the ones you developed are better than the best we have.
I would not necessarily put it that way. It is as good as any of the varieties that we have.
Okay.
Yeah, so the better part, saying that it is much better, and in terms of disease resistance, that one I can tell you, it's much better.
Okay.
And that is what you are looking for. In terms of the other qualities, it is as good as any of the best varieties that we have.
Wonderful, wonderful. And when we talk about cassava, I cannot fail to ask you about the situation where we have, because we have these things have been reported, we have children who went to a farm, they ate cassava, some of them actually developed complications, some of them died. I can't fail to ask you, what are the reasons that we normally have such cases?
So the cassava has a wide diversity. Actually, in the region, we have over 500 different varieties. There could even be more. So, and the variety has its own way of resisting against the enemies. These enemies could be the diseases, they could also be the insects, or they could even be wild animals.
Or even us.
Even us, you can be included in there.
Okay.
So, the varieties which are in the wild, if you eat, you die. So because they are bitter, and that bitterness comes because of the compound that cassava produces that is partly for protection. So the varieties that are developed, we develop varieties which are sweet. So that means that the component, which we call it carcinogenic potential, that means that they are capable of producing a compound that is similar to cyanide, that is toxic. So the bitter varieties, that compound is higher.
Okay.
The varieties that are sweet, that compound is way low, and it cannot affect you.
Okay.
But that said, the processing of cassava, so there are those varieties which you can go and pick in the field and even chew.
Yes, I've seen a lot of those cases.
Yeah. Some parts of the country, for example, if you go to Eastern, you go to the market and you see somebody just holding cassava and is chewing. Some people say it has its own advantages, the chewing of the raw cassava.
Yes.
So there's those compounds which are there. So, but the farmers know which ones and how to treat them. So if you grow the cassava that has those high levels of sanogenic potential, and you don't know, and you go and chew, or you go and process it in the wrong way, it may result to those, and that is because of diversity. And this does not necessarily, it's not only happen in cassava, it can happen in the crops, because when you're doing selections even in the wild, you eat varieties, you eat the ones that you can see even other animals are eating, because they have also known that this is not
good. True.
Yes, so the sanogenic potential in cassava is the one that results to that. But we test, as we do develop the new varieties, we develop varieties which have low levels.
Maybe before we talk about the cyanide levels in the new varieties, what about the cassava that are not ready for harvesting? Do they have the higher levels of these? Could this be one of the reasons? Because when children go, they may not actually be able to say this is ready for. Is that even a factor?
It could be a factor in some,
yes,
because as the plant matures, there are also the critical stages of when as I say that science has its own way of being able to balance. So there are stages at which the plant is affected most by a particular insect. And that stage, even when it is affected, it produces the chemicals produced to protect itself. So the production of those compounds, sanogenic potential, is also dependent on the environment.
Okay.
You might find one that is planted where it's too dry. Maybe that compound was also important in protecting the plants against drought. You may plant somewhere where there are a lot of insects. Yes. And that means that the insects have come to attack there are more insects that are coming to attack the plant. So that means that the plant might be producing more of those compounds. So what I was saying is that there are some stages in the plant development cycle when the plant is likely to be attacked by those pests more. And at that stage, so that means that it might be the potential of that compound might have increased a little bit. So, but now, when you're doing with developing new varieties, you also want to develop varieties that those spikes are not too high, to the extent that when you come and harvest at 6 months, if it can be able to produce at 6 months, that the compound is high. So you are looking at, yes, there is that you can say that at some point you have picked the variety when it is not fully mature. But there are also stages which you cannot go to harvest. For example, at that critical stage when you are saying that the potential could be very high, they may not having good roots to harvest and go and sell. And then after around 9 months, then you realize that the plant is at that level. The capability to produce those compounds also goes down, because it's also coming to the stage of when it's supposed to be dying anyway.
Okay.
So because you see, when you say that the crop takes 12 months, after the 12 months, even if you leave it, either the plant will die.
Natural
death.
Okay.
Or either the roots will start rotting, they'll start hydrolyzing, they'll start changing the composition, that it may not be of good or value to consume it again.
Okay.
So that means that you also have a window of when you can harvest. But the varieties that you are developing, even if there are those spikes which could be maybe having different changes, they will not reach the stage of where you can say that they're going to be deleterious to people.
Or they can harm them.
They can harm them.
Okay.
But that said, we still do have varieties which may have high levels. And especially the ones that are taking longer time. There are those which are taking 18 months to grow. Mm-hmm. And they have also their use. For example, if you go to Western Kenya, the people who are producing those varieties, they actually know how to process. You see them, those varieties will not be boiled. They'll harvest them, they'll go and ferment them, okay, or leave them to dry out in the sun. Mm-hmm. After that, then that compound is highly volatile. Once you expose it, when the plant is not growing and you expose it to the field, then and you are treating it in the right way, or even boiling, then you vaporize that compound. And that means that the product that is safe. So that means that variety, if you ate it when it was raw, it may have that bitterness, yes, and that compound. But when you process it, for example, exposing it to the sun as you dry, or as you boil, or as you ferment, then that compound, the effect is not there. So they know, farmers know the varieties they are growing, and especially those which are taking long time, and they know how to treat them. But the varieties that we produce now, there are levels which we say that we measure. we measure at the time of harvesting. We say that if it goes above these levels, then we don't think that it's good for release, especially if it's going for consumption.
Great. As we come close to the end of this recording, I would like to ask, there's those people, when we want to get these products now to the farmers, there are many other factors. And one of the factors is how this technology is perceived. The kind of misinformation surrounding genetically modified organisms and crops. Have you encountered misinformation in your area of work?
Oh yes, a lot of it.
How has it impacted your work? How would you say it has impacted you?
Our constitution gives people opportunity to air their views. That's public participation. And we have to inform them. There are those people who are saying the badness of the science. Some information is concerns which are good, and that's why there's risk assessment to make sure that some of those risks either perceived or real, and we have a scientific way of being able to, any risk must be assessed in a scientific way, in a way that you can say that you have assessed the risk and you have seen either the risk can be mitigated or the risk is not, it does not result to harm. So when you then you have listened to the people, and you have listened to other scientists, and they have said that these come up with systems of being able to assess some of those issues that could be assessed with risk. So the misinformation could be in different ways. There is that those which are involving risk, and risk could be as food to humans, as feed to animals, or could be to the environment, especially as the genes could be able to go outside and they can affect the other plants and become super weeds, or they all those factors come in. So there's the issue of safety that may come in there. There are others which may not necessarily be involved with the risk, and we call them socioeconomic considerations.
Okay.
And that may involve things to do, for example, with trade. So and issues to do with coexistence, issues to do with the seed sovereignty. So there are those people who are saying that when this technology comes, then the farmers will not be able to have the seeds, and it is being done, it is be owned by other companies that will be wanting to have benefits out of it. So for us, when we started working on cassava, because we are government institutions, and by nature of government institutions, we are doing it as a public good. So we are not necessarily saying that we are doing it as business. We don't aim to go there and recoup back the resources that we have used in science.
Yes.
Because this is resources came from the government, resources that came from donors, the resources that came from the people who wanted us to solve the problem. So we are not doing it as for business purposes. But once a crop has been produced, then you can go and use it for other. You can grow and sell the cassava the normal way that everybody else is selling it.
Mm-hmm.
So, but those misconceptions, those misinformation, and it has been a lot. You will see people saying that the genetic engineering is not a good technology, and then we have used it for 30 years in different parts of the country, of the world,
and in different fields, of course.
and in different fields. So, and all those misconceptions, those misinformations, saying that the genetically engineered crops, the seeds will not germinate, and so they are saying that you are being because you get the seed, you plant, then it's going to not germinate again. And especially for this cassava, it's once you can actually continue planting it over and over and over again. And we have done some plants we have planted for like 6-7 years, and in the field continuously, and coming and planting, and we see that it's still growing. The only thing is that when it's affected by all other factors, then you may need to have another cycle, a newer cycle plant that is not of having accumulated the bacteria and the fungi and all that, just like we do any other seed. When the farmer is going to get a cutting that is not sick.
Yes, so, of course.
Yeah. So the misinformation has been there, and we have tried to make sure that there's misinformation, there is lack of information, there is a need for people to understand,
yes,
and there is need for people to make decisions. And as a constitution implies is that there must be public participation. So we must give the people the information that is needed, so as they make the choices that they have, they're making when it is informed. So awareness creation is critical and it is important, as the people can know what is the truth about it. But it is there. It's big. It's not small.
Yes.
And we've had cases of people have gone to court, and of course we know, for example, now there is that case in the court that is saying that some people have gone to say that they don't want the crops to be GM crops to be released to the farmers.
Yes.
And so all those things are there. But the thing is that we keep informing the people what is the right information and what is true, what is not true, and so that then they can be able to make their judgment. The good thing is that the government is persuaded and they are aware. The people who are in the government systems, they have listened to us, they have seen what you have been able to do,
yes,
and they're given the go-ahead. But at the end of the day, it's the communities. It's not the government that's going to produce. It's the farmers who need it. And
yes,
we must be able to fulfill all the requirements as needed.
Finally, as we end, maybe you just look at that camera and tell those people who have reservations based on the limited knowledge or information they have about this technology. What can you tell them?
What I would say is that this is one of the highly tested, evaluated, and assessed technologies. Any variety that goes to the farmers, any product that goes to the market for use has been ascertained at the highest level possible. So whether they are the seeds, the quality of the seeds, and how the seeds can be able to grow to the all of that information has been the government institutions and everybody else who is involved in the process has been convinced that on the safety and the good quality of this and what the technology can give. So what I would want to tell to the farmers and consumers that there is nothing to fear. This science, it is us, it's us as a part of the local communities. I know it is me who will be consuming it. It's my children, it's my parents, it's my sisters and brothers, it's my neighbors. So, and whatever I do it for the common good. And this is not only for me, it is a community world over. And if you see all other organizations have accepted and have agreed that it is true. And the history of use over 30 years of technology with no cases of where we are saying that it has necessarily been linked to the technology. What we are saying is that we should have no fear, we should have no concerns, because all those things have been evaluated. And you have the best assurance from me as a scientist, and to say that it is actually safe technology.
Wonderful, wonderful. What a way to end it when you give that level of assurance that you are developing it. It's for your children, it's for people close to you, and there's no way you're going to develop something that could be harmful to them.
Exactly.
Folks, you've heard it from one of the best minds in Kenya and one of the lead scientists who actually led here in Kenya, and they were the first ones to develop genetically modified cassava. That just tells you how good we are as a country in terms of capacity, human capacity to develop these things. And even when you move around the globe, you will find more scientists who are from Kenya and they have this ability. So it's just that we are exporting these minds, these brilliant minds, and now we have one that has actually developed something for us that we need to consume, not just here in Kenya, but across Africa. So thank you very much, Prof.
Thank you.
We appreciate your time. So we'll be joining you maybe the field one of these fine days to see this and also interact with the crop and see how best that is coming along. So thank you very much.
You're welcome. I appreciate it.
Thank you. So, folks, that is it from here. We've heard that we were created to subdue, and to recreate. We were given that ability. So we are not playing God when scientists are doing these things. So thank you very much. Till next time, this is Ezekiel Nganga, and this is Africa Science Dialogues.